Hello and welcome back. So this mini lecture is going to be on the physiology of tension production. So we're going to first take a look at how tension is produced in the individual muscle fibers, and then we'll take a look at how tension is produced in the whole muscle, kind of the summation of all of the individual muscle fibers. So remember, when we talk about tension is like lifting something heavy, right? So you're lifting up something heavy, you have to produce tension to overcome the weight of the thing that you're lifting. So that's what we call the resistance. So tension in muscles has to be high enough to overcome the resistance of what you're trying to lift up if you want to move that item. You can still produce tension even if you don't move the item. We're going to talk about different types of whole muscle contractions where that might come into play at the end of this video. So just to recap, you should have gone through the physiology of muscle cell contraction. So that's the excitation contraction coupling. So have a good kind of grasp of what's going on to cause the muscle cells to get smaller, the interactions between the thick and thin filaments, the shortening of the sarcomeres, myofibrils, muscle fibers, fascicles, whole muscle cells, all that kind of stuff, right? All right, so when we take a look at tension production, in the muscle fiber, we're going to do muscle fibers first. There's two factors that come into play for how much tension is produced in an individual cell. So first is what we call length tension relationships. How much overlap do we see in the sarcomere when we start the contraction? That's the length and tension relationship. And then the other factor is how fast is that action potential showing up in the neuromuscular junction? So is it just one signal, and then that's it? Or are your neurons firing, is that going to affect the amount of tension that individual muscle fiber can produce? So we're going to answer those questions. That first thing we're going to take a look at is the length tension relationships. So what we have here is a graphical representation of the tension production represented as percent of maximum. So if we measure the maximum amount of tension that can be produced by a muscle, then that is going to be 100%. If it can't produce any tension, 0%. So we're not putting a weight to it. It's just percent of maximum. So there is a optimal length of your sarcomere, kind of right in this short little zone of this length tension relationships. That's going to be your optimal length of your sarcomere between Z disc to Z disc of when that contraction starts, you're going to reach that peak 100% tension production. But we have a normal range, right? So we're going to kind of stay within a little bit on either side of that optimal. But I'm going to first start by taking a look at the extremes. Let's take a look at the extreme over here. 0% tension, and you don't need to know the units like the micrometers here, just more of what's happening. So when your zone of overlap is 0, right? So here's your zone of overlap. When there's no overlap, when that signal comes in and the thin filaments or the thick filaments are trying to reach up and grab the thin, they're not going to be able to. So there's no contraction because your interaction isn't happening. You have to have some overlap for those thick filament myosin heads to grab onto the thin filament G-actin proteins and move them towards the M line. So if we are to pull and stretch our sarcomere so far apart that the zone of overlap is lost, we're not going to get any tension production. Okay, so now let's come over here to this extreme over here. If your zones of overlap are big, right, but we moved our Z lines so close together that your thick filaments are bumping up against your Z discs, and you have no more I bands and it's just A band and the Z disc is right up next to the A band, even if you have all of that zone of overlap, so lots of zone of overlap, you can't make it any shorter. I can't bring my fingers any closer together because they're already as close as they're going to be. So if your sarcomeres are really close together when the signal arrives, you're not going to be able to produce any tension. Okay? In these two extremes, you're not going to produce any tension. But fortunately, our bodies don't really go to those extremes too much because we can strengthen our arm out, but I'm still going to have zones of overlap. If I were to break my arm, and my forearm pulls, it's like being pulled in a really bad accident, I might be able to pull my muscles so far that I'm not going to be able to contract them. Or I can't really, just the nature of my connective tissue and my muscle, I can't contract my muscles so much that my sarcomeres are going to be too short. So these extremes don't really happen in real life. We are more likely to kind of hang out in this optimal normal range where we have some overlap to have actually interaction, but we also have some distance between the thick filaments and the Z discs to allow for tension to actually be produced. Okay, so that's one factor. And this factor doesn't really change much. So it's kind of set based on the anatomy of your body. Our skeletal system, the connective tissues, all those mysiums of our muscle do not allow us to either be stretched too far or, contracted so much. So this isn't really a day-to-day issue or minute-by-minute issue to tell us how much tension can be produced by individual muscle fibers. So it is a factor, but it's not one that changes very much unless you hit some extreme situation. The more flexible or variable factor of those two is the frequency at which the stimulus arrives at the muscle. So we're going to start with the stimulus, just one stimulus showing up at a muscle, and see what happens. So again, here's our tension, percent of maximum. And then on the x-axis, this is time. So we're going to be dealing with milliseconds. Okay. So let's say at time 0, your stimulus shows up. So this stimulus is the arrival of acetylcholine in the neuromuscular junction. That's our excitation part, right? So the green arrow is showing you arrival of acetylcholine. There's a tiny little span of time, about 2.5, 3 milliseconds. That's so fast we can't even comprehend that amount of time. It's called the latent period. And the latent period is basically measuring all of the events of the neuromuscular junction, the arrival of the acetylcholine, traveling down the T-tubule, release of the calcium, binding to the troponin, moving the tropomyosin out of the way, the binding of the myosin head, then tension starts. So all those things of that coupling that we learned in the other physiology video happens in about 2.5 to 3 milliseconds. That's pretty amazing. Then we get contraction. Right, so our contraction is happening. Contraction, contraction, contraction. And so we have maximum tension produced. The stimulus goes away. Acetylcholine esterase removes the acetylcholine. The calcium is getting pumped back into the sarcoplasmic reticulum. Active sites being covered by the tropomyosin. Myosin heads let go. Muscle is being relaxed. Relaxation. So from stimulus to relaxation in a typical muscle, or this is the gastrocnemius, which is your big calf muscle, takes about 40 milliseconds. Okay, so 1,000 milliseconds is 1 second. So this is really, really fast. So that's one twitch. And we can see your eye muscle, here's the gastrocnemius, your soleus is another calf muscle. So there's different lengths of time. So if you ever have a twitch, like your eye muscle kind of goes ping, 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 it's doing its whole thing in like 15 milliseconds, okay? Where the soleus might be over 100 milliseconds. But when you're talking about work to be done, a twitch is not a very good movement. You can't lift stuff with a twitch. It's not producing enough tension to overcome any kind of resistance. But we have to define what it is, because what we're going to actually do is start adding some of these together when the stimulus show up quicker. The frequency increases if this stimulus, that acetylcholine, is showing up faster than a full twitch, okay? So this is that other factor, frequency of stimulation. How fast are those stimuli showing up? So again, the green arrow is representing arrival of acetylcholine in the neuromuscular junction. So if we do a full cycle of contraction and relaxation, right? So what I'm highlighting right here is one twitch. That slide we just looked at, that's it. So we have contraction and relaxation, full relaxation to like back to 0. Then another stimulus shows up. We get another twitch, okay? All the way down to relaxation. Another stimulus shows up, we get another twitch. So basically there's just a whole bunch of twitches added together. And at every subsequent twitch, even though we're letting the muscle come all the way back down to rest, we increase the tension just a little bit. And this is what we call TREP. So TREP kind of means like steps or stairs. So TREP, we can't get to the maximum tension, that 100% tension here. It's not even close. But this is the maximum tension we can produce in TREP when the muscle cell can relax, completely relax in between each stimulus. Now the next one is, what if we're starting our stimulus and we're starting our relaxation, or we're starting a relaxation phase, and another stimulus shows up before we have a chance to completely relax. So here we're going to start another contraction cycle, and then we start to relax, but then another stimulus shows up before we can relax. And so this is a pattern we get, which is called wave summation. And you can see we can produce quite a bit more tension in wave summation than we could in TREP, because we're triggering the muscle to contract before it has a chance to completely relax. Okay, let's speed that up. Let's make it come a little bit faster. So in incomplete or unfused tetanus, what we see is the stimulus is showing up even faster. The muscles reach its peak. It's starting to relax, but then it just gets hit again before it gets to relax even more. And so this is what we get is incomplete tetanus. We get more tension than we could in wave summation, but we're still not reaching maximum contraction or maximum tension. That comes in what we call complete or fused tetanus. Here, the stimuli is arriving so fast that the muscle fiber is not even allowed to go into any relaxation phase. That neuromuscular junction is just firing, acetylcholine, 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 and the cell just stays contracted, right? We don't have any relaxation, so it just stays short. There's no variation, there's no relaxation, there's no nothing. In reality, we don't really hit complete tetanus very often. It can happen in the disease tetanus or some other neurological disorder where your motor neurons are firing too quickly. The disease tetanus is caused by a toxin that it interferes with your motor neurons and keeps them from turning off. So you're just constantly telling your skeletal muscles to contract, and so that's why you get tetanus, the disease, which is horrible. So get your vaccine. All right. In reality, we are more likely to be a combination of these frequencies. We don't really see this too often in reality, in a day-to-day activity. So depending on what your body needs, it is going to determine your frequency of muscle contraction. So for example, if I'm going to pick up a pencil is not very heavy, and my brain knows from my life that I've lived that a pencil doesn't weigh very much. And so when I go to pick up a pencil, I'm not going to need to stimulate my muscles at incomplete tetanus to produce a whole bunch of tension to pick up a tiny pencil. So I might stimulate my muscle fibers maybe in TREP or wave summation frequency, a little bit slower of a signal, to produce enough tension to pick up a pencil. I don't have anything really heavy, but what if my binoculars, I have binoculars here, what if these are really heavy? And I know they're really heavy because I've experienced them before, and I go to pick up my binoculars, I might have to stimulate my muscles at incomplete tetanus frequency to make enough tension to lift up the heavier item, okay? So that's the relationship between frequency, how fast are those stimuli showing up, to how much tension individual muscle fibers can produce. Okay, so that's tension at the individual muscle fiber. Length frequency or length tension relationships, which is kind of set by your connective tissues, doesn't really change, but this is the factor that's going to change the most for muscle fiber tension. So now we have to take a look at, we don't lift things up with one muscle fiber, we lift things up with whole muscles. So how do we know how much tension is produced by a whole muscle? Well, first of all, we have to see how many of those cells are actually being stimulated. So I'll try to, maybe I'll highlight with my blue ink. So let's highlight these blue, motor unit number 2. A motor unit is a single neuron and all the muscle fibers it innervates. So my blue motor neuron sitting there in the spinal cord is going to send out its axon, and then its terminal branches are going to go and innervate 1, 2, 3, 4, 5, 6, 7 muscle fibers. So when that blue motor neuron fires, all 7 will react the same. If it's at wave summation, the frequency of my blue neuron, all of those blue muscle fibers will contract at that particular frequency and at that particular tension production. So if I need to only pick up something light with all my muscles, right, I might only activate 1 motor unit. But what if I got to pick up something heavy, right, my heavier object? Blue motor unit at wave summation might not be enough. So maybe I'll activate red motor unit, and now I'm going to turn on blue and red, which means my 7 of my blue and 1, 2, 3, 4, 5, 6. So now I've got 6 plus 7, 13. I've got 13 muscle fibers. Maybe being stimulated at incomplete tetanus is going to be enough for me to produce the tension needed to lift this heavier object. Right? If I need to lift binoculars made out of lead, right, that's probably not likely, but maybe I might have to turn on my purple motor unit and add these muscle fibers, all maybe firing at incomplete tetanus frequency for me to lift something that's really heavy. So your brain, you don't really have to think about it. All you have to do is think about what you want to lift up, and your life experiences, that observation tells your brain how many motor units you need to activate and at what frequency you need to activate them. Are you activating all 3 motor units at incomplete tetanus? Well, that might be something very heavy that you need to pick up. Or you're activating only 1 motor unit at TREP, and so maybe you're only picking up a card or a piece of paper, something light like a feather. So all of that knowledge is gained by your life experiences. What if you do go pick up a pencil that looks like a regular pencil, but it's made out of lead? Your brain's gonna go, what? Because you're expecting to only pick up a pencil that may weigh a gram or 2, but if it is made out of lead and weighs a whole pound, you're gonna try to pick it up and you're gonna be like, oh, I can't pick it up with that expected motor units and frequency combination. Excuse me. Even though you're not thinking that. So then you're going to have to reconfigure your signaling, and your brain's going to have to work on the fly to say, oh, this pencil is way heavier than a normal pencil, and you'll have to activate more motor units, and that is what we call recruitment. So recruitment is you're bringing in more, activating more motor units to produce the tension you need to overcome the resistance that you're presented with. So this other picture here, so what if, right here, what if we're talking about postural muscles, not lifty muscles like our biceps or something, but just standing up straight muscles? We don't want to fatigue any muscles completely. And so what we need to do is we need to kind of let them be on in a rotating basis. So we might have an average tension production on the tendon holding up some of our postural muscles, but it's the motor units taking turns maintaining that tension. So motor unit 1 gets to make the tension, and then it gets to rest, and then it comes back for its turn. But in the meantime, motor unit 2 had a turn, motor unit 3 had a turn, and then each one gets to rest before they're called upon to maintain tension in, say, like a big back muscle or a leg muscle for standing upright, or your neck muscles to make sure you're not, floppy over during the day. When you're sleeping, it's okay to be floppy over. Okay. So we did that. Okay, so the next thing we're going to talk about is motor units, muscle tone. So I always show this picture of Arnold Schwarzenegger, right? The Terminator, or Conan in this picture. So muscle tone is our background level of muscle activity. Not that we're actively using our muscles, but I would guess that in the picture on the left, he's actively flexing for that picture. But he could probably just relax his muscles at that age when he's young Arnold, and he would still look kind of buff. What that means is he has a high muscle tone. His muscles are at a higher level of partial background level of activity. Now, as you age or as you don't work your muscle tone goes lower. And so they look a little bit softer and a little bit flabbier because your muscle tone is low. If you want to burn calories, like if one of your goals are to lose weight or to burn calories, the best way to do that is increase your muscle tone. So weightlifting exercises, cardio is good for the heart and your lungs, But weightlifting exercises increases the ability for your muscles to be burning calories in the background while you're just resting and sleeping if your muscles have a higher muscle tone. So that's a good goal. It's good for your bones, right? Because we want to have good, strong bones, and it's also good for your muscles to keep them with a good muscle tone. All right, the last couple ideas are with whole muscle contractions is when we contract a muscle, are we able to overcome the resistance with our tension production or not? If we are, we call these isotonic contractions. An isotonic contraction is when you produce enough tension that your muscle changes length. And there's two types of length change. One, your muscle gets shorter, right? So that's the first picture. So I have a 2 kilogram weight. I'm lifting the weight. So pretend this is my 2 kilogram weight, my pencil. So I'm lifting my weight and my tension can overcome that resistance and my muscle is getting shorter, right? So it's like this, and then it's getting shorter. That is called a concentric isotonic contraction. The tension is kind of staying the same, but the length is changing. Now, when you are doing weight-bearing exercises, so I'm going to stand up a little bit. So if you're in the gym and you're like, okay, so I'm going to lift my weight, are you supposed to just like let go and let it drop down? No, you're not. You're supposed to lift and then slowly lower it down, right? So lift, that's concentric, and eccentric. Are you still producing tension? Yeah, you're still producing tension, but your muscle is lengthening. So this is what we call an eccentric contraction. So you are producing tension, but the muscle is getting longer. It's just the resistance is more than your tension that you can produce. Okay, so concentric is kind of getting shorter, eccentric is getting longer, but in both cases you have tension being produced. And then lastly, we have isometric contraction. So isometric is you're producing tension, iso meaning the same, metric meaning length. You're not increasing the length of the muscle. So again, if I'm going to pull up on my table, like I'm producing tension, my muscle is contracted, I can feel it, but I'm not lifting up my table. I don't have enough strength to overcome the resistance, but that doesn't mean I'm not producing tension. So in that case, the length of the muscle stays the same, but the tension, you can produce tension, but the length of the muscle doesn't change. So in this case, the 6 kilogram weight is too much for the muscle to overcome. So here you're producing tension, you just cannot lift the load, okay? All right, so that is our tension production mini lesson. So we covered tension production in individual muscle fibers and then tension production in whole muscles. All right, I will see you next time for ATP usage in the cell. Bye.